So I want you to imagine that you're carrying around a half-gallon container everywhere you go, like literally everywhere.
Yeah.
And it weighs, well, exactly as much as your brain, roughly 1 kilogram.
Right.
And here is the really unsettling part. It is entirely filled with invisible living creatures.
It's terrifying when you phrase it like that.
Right. But it's true.
It is. And, if you drop that container, you wouldn't just get a little sick. You'd basically cease to function entirely.
Wow.
Yeah. You'd lose the ability to extract energy from your food. You'd lose the capacity to regulate your immune system. And you would rapidly lose the ability to produce the neurotransmitters that keep your brain from, like, spiraling into a panic attack.
It's a completely wild thought, but it is the absolute biological reality of what it means to be a human being today.
Mm.
We walk around thinking we are, entirely in charge, that we're this autonomous organism, but biologically, we are completely outnumbered.
Oh, massively outnumbered.
I mean, for every single human cell in your body, there is a microbial cell. It's a 1-to-1 ratio, but it's the genetics that really shatter the illusion of control for me.
The gene ratio is staggering.
It is. For every one human gene you possess, there are at least 360 microbial genes actively functioning inside you right now.
Yeah, 360 to 1.
Yeah.
We have effectively outsourced our biology.
Outsourced it.
Yeah, exactly. Over 2 million years of evolution, we didn't develop all the metabolic machinery we needed to survive because, well, it was far more efficient to just host organisms that already possess that machinery.
That makes total sense. Like, why build the factory when you can just hire the workers who already have the tools?
Precisely. The scientific community has largely stopped viewing these microbes as, like, hitchhikers or passengers.
Right.
They're universally considered a second organ.
A second organ.
Yeah, an organ that is as vital to your survival as your heart or your lungs. But the crucial difference is this organ is completely malleable.
Malleable. And that malleability is exactly why we are doing this deep dive today. Because we spend so much time obsessing over diet culture.
Oh,
People argue fiercely over strict keto or veganism or, paleo or counting macros. But if you actually look at the mechanics of digestion, we are putting the cart before the horse.
We really are.
We are obsessing over the exact type of fuel we are putting into the machine, completely ignoring the fact that if the microscopic pit crew inside you isn't equipped to process that specific fuel,
You're going nowhere.
exactly, you are going nowhere. The food doesn't matter if the microbiome is broken.
That right there is the crucial paradigm shift.
Yeah.
You cannot decouple human cellular health from microbial health.
Right.
If you ignore the vast majority of the genetic material operating inside you,
Mm-hmm.
you're ignoring the primary drivers of how you feel, how you digest, and how you fight off chronic disease.
So the mission for today's deep dive is that we need to completely map out this ecosystem.
Yes, let's do it.
And I really want to avoid the trap of just saying the buzzword gut health.
I'm so glad you said that.
Because the gut is just one neighborhood, right? If we are going to understand how these invisible armies act as, like, our ultimate detoxifiers, how their hidden digestive enzymes work, and how we can actually fortify them, we have to view the human body as a planet.
A planet with wildly different, distinct biological zones.
Exactly. So what is the most logical place to start mapping this planet?
The outermost surface, the skin.
The skin.
Yeah. Most people think of the skin as a static, waterproof wrapper meant to keep the inside in and the outside out.
Like saran wrap.
Right, like saran wrap. But it is a highly dynamic, exposed battleground.
And it's not just one uniform environment across the whole body, is it?
Not at all. It is incredibly diverse. The skin provides highly distinct physical, chemical, and biological terrains.
Okay, like what?
Well, you have arid, dry zones, like your forearms. You have humid, sweaty zones like your underarms. And you have lipid-rich, oily zones, like your face and your scalp.
Right.
Across these diverse terrains, human skin hosts 48 distinct bacterial types, alongside various fungi and viruses.
Okay, let's make this tangible for a second. Give me an example of what is actually living and, working on my face right now.
Well, one of the most fascinating residents is a fungus called Malassezia.
Malassezia.
Yeah. It lives predominantly in your sebaceous glands. Those are the glands that produce the oil or sebum on your skin.
Okay, so it lives in my oil glands.
Right. And Malassezia literally feeds on those human oils. Now, when the ecosystem is perfectly balanced, This fungus is harmless. It might even help break down cellular debris.
Good to know.
But if your skin chemistry changes, or if your immune system dips, melanocytes can overgrow.
And what happens then?
When it overgrows, especially on the scalp, the byproduct of it feeding aggressively on your oils creates an inflammatory response. That response causes your skin cells to flake off rapidly.
Wait, are you describing dandruff?
I am. Dandruff is not just dry skin.
Oh, wow.
Yeah, it's an ecological imbalance. A specific fungus has been allowed to aggressively overconsume your sebum.
That completely changes how I view washing my face or my hair. I mean, if my skin is a thriving ecosystem balancing 48 different types of bacteria and fungi,
Which it is.
then when I jump into a hot shower and scrub myself down with harsh antibacterial soaps, I'm essentially wiping out an entire rainforest just to feel clean.
You are actively clear-cutting the forest. And the problem isn't just the removal of the microbes, it's the alteration of the chemical environment itself.
How so?
Healthy skin bacteria, like Staphylococcus epidermidis, are highly protective. They actually produce antimicrobial substances to naturally kill off invasive, harmful pathogens.
Wait, like they make their own antibiotics?
Exactly. They produce compounds like bacitracin, which is literally what you'd find in an over-the-counter antibiotic ointment.
That's incredible.
Furthermore, they help maintain what we call the acid mantle of the skin.
The acid mantle. Okay, meaning our skin is naturally supposed to be acidic?
Yes. Slightly acidic, hovering around a pH of about 5.
Okay.
This acidity is a very hostile environment for many dangerous pathogens. But when you use heavy alkaline soaps, which most commercial soaps are, you strip away those beneficial bacteria and you neutralize that acid mantle.
So you are effectively opening the gates of the fortress.
Yes, and inviting invasive species to set up camp on your skin.
Okay, so if the skin is this protective outer layer, what happens when something goes wrong deep inside the fortress? Because I know a lot of people who suffer from chronic skin issues, right? Eczema, psoriasis, mysterious rashes.
Very common, yeah.
And they spend thousands of dollars on topical creams that never really seem to fix the root issue. It always comes back.
That brings up a critical physiological mechanism. The skin acts as a diagnostic mirror for internal toxicity.
A mirror.
Yeah. Your body has primary pathways for eliminating waste and toxins, mainly the liver and the gastrointestinal tract.
Yeah.
But if your gut is compromised or your liver is overwhelmed by a heavy toxic load, the body still has to get that waste out somehow. It has to go somewhere,
So it defaults to the secondary elimination organ.
which is the skin.
Wow.
Precisely.
All right.
The body physically pushes inflammatory waste products out through the skin, so that rash or that eczema flare-up is rarely a localized skin problem.
It's an internal problem.
It's a systemic alarm bell. It indicates that the gut's detoxification load has exceeded its capacity. You can apply all the topical steroids you want to suppress the local immune response on the skin.
Just putting a Band-Aid on it.
Exactly.
Yeah.
But until you fix the internal microbial ecology, the fire will keep burning.
It makes sense that the outermost layer is a harsh, reactive environment. But if we move inward past the protective skin, the vulnerabilities are the openings, right?
Right.
The mouth, the respiratory tract. I know from the sources we reviewed for this deep dive that the oral cavity hosts around 56 different bacterial types and the esophagus has 43. But what truly fascinates me about the establishment of these internal environments is how it all begins. Day one.
Oh, yes.
The sheer biological stakes of how a human being is actually born.
There is perhaps no more critical moment in the development of the human immune system than the journey through the birth canal.
Really?
Absolutely. The vagina hosts 5 main types of bacteria, heavily dominated by species like lactobacillus. As a mother approaches full term, her vaginal microbiome actually shifts. It thickens and changes composition, specifically preparing to inoculate the infant.
Inoculate. So the baby is basically taking massive gulps of bacteria on the way out.
Exactly. The baby is born essentially sterile in the womb. As it passes through the vaginal canal, it is coated in and swallows this dense microbial cocktail.
Wow.
This is the pioneer bacteria. It enters the infant's digestive tract, takes up residence, and immediately begins communicating with the infant's naive immune system.
Teaching it.
Teaching it what to attack, what to tolerate, yes.
But hold on. If that vaginal inoculation is the evolutionary baseline for a healthy immune system, what happens during a cesarean section?
It's a completely different story.
Right, because we are talking about pulling a sterile baby out through an incision in a highly sanitized surgical theater. They completely bypass the mother's pioneer bacteria. Are C-section babies fundamentally disadvantaged from day one?
They start with a profoundly different ecological baseline, and the physiological consequences of that are very well documented.
Oh, what happens?
Instead of being colonized by the mother's vaginal flora, a C-section infant is primarily colonized by whatever happens to be floating around the hospital room,
Like what?
mostly the bacteria on the skin of the surgeons and nurses,
Hmm.
primarily Staphylococcus species.
Oh, man.
Because they lack that initial intense education from the mother's pioneer microbes, their immune system doesn't calibrate properly.
Can you explain that mechanism? Like, how does missing one dose of bacteria at birth lead to an uncalibrated immune system?
Think of the immune system like a massive standing army of young, aggressive recruits.
Okay.
If you don't give them a clear enemy to fight, or proper training on who the civilians are, they get bored and paranoid.
That's dangerous.
Very. The lactobacillus from a vaginal birth acts as the drill sergeant. It regulates the immune system, telling it, hey, this protein is food, ignore it. This particle is dust, ignore it.
Right.
Without that early microbial training, the immune system becomes hyperreactive. It starts attacking harmless things.
Which manifests as allergies.
Exactly. This is why epidemiological data consistently link C-section births with a significantly higher future risk of asthma, food allergies, type 1 diabetes, and even obesity.
That is wild.
The foundational programming of the immune system was completely bypassed. Now, this doesn't mean C-section babies are doomed forever.
Right, there's a way to fix it.
Yes, the microbiome can be cultivated and repaired over time through breastfeeding, environmental exposure, skin-to-skin contact. But it starkly illustrates that these invisible creatures are the primary architects of our early biology.
So we've covered the outer shield, the entry points, and the pioneer settlement. Now we have to trace the path down into the actual engine room, right? The gastrointestinal tract.
The GI tract, yeah.
And what I find so interesting is that the population density of these microbes doesn't just like gradually increase as you move down the tube.
No, it fluctuates wildly.
Right. It changes drastically based on the extreme chemical conditions of the local neighborhood, starting with the stomach.
The stomach is essentially a biological incinerator.
I love that phrase.
It really is. It is a vat of highly acidic hydrochloric acid with a pH that can drop down to 1.5.
Wow.
To put that in perspective, battery acid has a pH of around 1.0.
So it's almost as corrosive as battery acid. Why do we need it to be that acidic? Is it just to break down a tough piece of steak?
Breaking down proteins is part of it for sure,
Yeah.
but the primary evolutionary function of that extreme acidity is sterilization.
Sterilization.
Okay. The stomach acts as a brutal gatekeeper. Every time you eat a piece of fruit or a handful of nuts or a piece of meat, you are ingesting millions of environmental bacteria.
Right, they're covered in bugs.
Exactly, and many of those could be highly pathogenic. The hydrochloric acid, combined with bile salts, which act like biological detergents to tear apart bacterial cell walls.
Oh, wow.
Yeah, they destroy the vast majority of these microbes before they can ever access your vulnerable intestines.
Which means the few microbes that actually do live permanently in the stomach must be incredibly tough.
They are highly specialized extremophiles. Because of that hostile environment, you only find about 10 to 1,000 bacterial cells per milliliter in the stomach.
That's tiny.
It's a microscopic desert.
But then you leave the stomach, you pass through the small intestine where, nutrient absorption happens, and eventually the flow slows down. You enter the large intestine, the colon.
And everything changes.
Suddenly, that desert turns into the most densely populated ecosystem on the face of the earth.
The shift in density is absolutely staggering. By the time you reach the colon, the population explodes to somewhere between 10 billion and 1 trillion microbial cells per milliliter.
1 trillion per milliliter? Why? Why do they all congregate at the very end of the digestive tract?
Because they're waiting for fuel.
Fuel.
The stomach broke down the proteins, the small intestine absorbed the simple sugars and the fats. But there's a massive component of our diet that human biology is completely incapable of breaking down.
I just can't process it.
We lack the genetic instructions. We don't have the enzymes. So it passes intact all the way down to the colon, where this trillion-strong army is waiting to feast on it.
And that fuel is fiber.
Yes, fiber is the absolute foundation of human health, and unfortunately, it's the most misunderstood nutrient in the modern world.
Let's completely reconstruct how we think about fiber right now, because the marketing around it is terrible.
It really is.
We've been told for decades, drink this gritty orange powder, eat your bran flakes, it's a digestive broom. The narrative is that fiber is just indigestible roughage that physically sweeps through your intestines to keep you regular.
Right, like a scrub brush.
Yeah, but it's not just structural debris, is it?
Not at all. Treating fiber like a simple digestive broom is like treating a highly complex computer network like a simple abacus.
Good analogy.
Dietary fiber consists of highly complex, non-degradable carbohydrates. These are intricate structural molecules from plants. When these fibers reach the colon intact, the beneficial bacteria ferment them.
Fermentation, like how yeast ferments sugar to make beer or alcohol.
Very similar mechanism, yes, but with entirely different outputs. The bacteria consume the complex fibers, and through the chemical process of fermentation, they produce entirely new metabolic byproducts.
Yeah, and you need those.
We desperately need them. The most critical of these byproducts are called short-chain fatty acids, or SCFAs.
Okay, let's get specific here because from the material we looked at, these SCFAs are basically the secret currency of the entire microbiome. We are talking primarily about acetate, propionate, and butyrate.
Right.
And they're produced in a very specific biological ratio, right? 1 to 1 to 3. But I don't want to just list these off like we're reading a textbook. What does a molecule like butyrate actually do when it hits my colon?
Let's look at it from the perspective of a single human cell lining the wall of your colonocyte.
A colonocyte, okay.
Every cell in your body needs energy to function. Most of your body runs on glucose from your bloodstream. But your colonocytes are unique. They derive up to 70% of their total energy directly from butyrate.
Wait, so my human colon cells don't get their energy from the food I eat. They get their energy from the waste products of the bacteria eating the food I eat.
Precisely.
That is wild.
It is a stunning evolutionary trade-off. We provide the bacteria with a warm, oxygen-free environment and a steady supply of complex plant fibers. In exchange, they ferment those fibers into butyrate, which physically powers the cells lining our gut.
So what happens if there's no butyrate?
If you do not produce butyrate, your colon cells literally begin to starve. They atrophy, and they undergo apoptosis, which is cellular death.
That is incredible. And what about the other two? Acetate and propionate. What do they do?
Once they're produced in the colon, they absorb into the bloodstream and act as systemic signaling molecules.
Oh, so they leave the gut.
Yes. Acetate travels to the liver and the peripheral tissues, where it plays a major role in regulating fat accumulation and improving insulin sensitivity.
So it helps with metabolism.
Huge for metabolism. Propionate goes directly to the liver to inhibit cholesterol synthesis, and it also acts on the pancreas to enhance the function of beta cells, which is what helps control your blood sugar levels.
So let me get this straight. If I eat a piece of broccoli, the human part of me can't really digest the fiber. The bacteria eat it, they turn it into these fatty acids, and those fatty acids circulate through my entire body to tell my pancreas to regulate insulin and tell my colon cells to stay alive.
Exactly. It's a perfect symbiotic loop.
But this raises a terrifying question.
Right.
What happens when we break the loop? Like, what happens if I just stop eating fiber?
If you stop feeding them fiber, you initiate a biological catastrophe.
A catastrophe?
Truly. These bacteria are living organisms, right? They're driven by the imperative to survive. If you cut off their primary food source, they don't just politely pack up and die.
They look for food.
They look for an alternative food source.
And what is the alternative?
Your own body.
What?
Specifically, the thick, protective layer of mucus that coats the entire inner lining of your large intestine.
Hold on. You are saying that if I don't eat enough fiber, the bacteria inside me will literally turn on me and start consuming my own internal organs.
That is the biological reality. The mucosal layer is composed of glycoproteins called mucins. In the absence of dietary fiber, certain species of gut bacteria will shift their metabolic pathways, they deploy different enzymes, and they begin foraging on these mucins.
Foraging on my mucus layer.
They literally eat away the mucus lining of your colon just to stay alive.
That sounds like a sci-fi horror movie. But let me push back on this a little bit. If this massive bacterial army starts aggressively eating my intestinal wall, why doesn't my immune system immediately recognize them as the lethal threat and just carpet bomb the entire area? Like, why do we tolerate this mutiny?
Well, your immune system does recognize the threat. But you have to understand, it's a structural failure, not just a standard infection. That mucosal layer isn't just there for lubrication, it is a vital physical barrier. It keeps the trillions of bacteria, the undigested food particles, and the environmental toxins safely inside the tube of the intestine, away from your bloodstream.
Right, it's the wall.
When the bacteria eat away that barrier, the intestinal wall becomes highly permeable, We call this leaky gut.
Leaky gut. So the physical wall is breached.
Exactly. And when that wall is breached, bacterial fragments and toxins begin slipping into the bloodstream. Now your immune system reacts.
It panics.
It panics. But it can't just carpet bomb the colon without destroying the host, which is you. Instead, it launches a low-grade, chronic systemic inflammatory response.
Just constant simmering warfare.
Yes. And this is the physiological root of chronic fatigue, autoimmune flare-ups, joint pain, and metabolic dysfunction. Your body is perpetually fighting a microscopic war at the border wall because you starved the guards.
Wow. So if anyone listening has ever gone on an extreme long-term diet, like a strict zero-carb carnivore diet, or even just a highly processed Western diet devoid of real plant matter, and after a few months they feel sluggish, inflamed, or physically terrible, this is the mechanism.
This is exactly what's happening.
They have starved their microbial workforce, the workforce ate their gut lining, and now toxins are leaking into their blood.
And it actually goes even deeper than that. This brings up the issue of long-term microbial extinction. Extinction. If you maintain a severely low-fiber diet for years, you don't just suppress the fiber-digesting bacteria. You can lose those specific strains entirely. They go extinct within your unique internal ecosystem.
Oh, which totally explains why people who avoid plants for a year suddenly try to eat a bowl of beans or a salad and experience agonizing bloating and pain.
Exactly. They assume, oh, my body can't handle plants. Vegetables are toxic to me.
Right, you see that on the internet all the time.
All the time. But no, vegetables aren't toxic. You literally no longer possess the biological machinery, the specific bacterial populations required to break down those complex fibers.
Because you starve them to death.
Yes. So the food just sits there and ferments improperly. It produces excessive methane and hydrogen gas instead of the beneficial SCFAs.
Yeah.
You have to meticulously, painfully rebuild that ecosystem over months.
To really drive home how far we've fallen from our evolutionary baseline, let's look at how people eat when they are actually connected to their food sources. There was a landmark piece of research that perfectly illustrates this, comparing the microbiomes of children living in a rural village in Burkina Faso, Africa, to children living in Florence, Italy.
Ah, yes, the Lionetti study. It is one of the most revealing snapshots of how modern industrial diets have decimated our internal ecology.
Let's expand on this. What does the daily life and diet of a child in rural Burkina Faso actually look like, and how does it translate into microbial health?
Well, these children are eating a diet that closely mirrors early human agriculture. They consume large amounts of complex, tough carbohydrates.
Like what kind of foods?
Things like millet grain, sorghum, black-eyed peas, and various locally foraged vegetables. Their diet is predominantly plant-based and incredibly high in structural fibers. They're easily consuming 30 to 40 grams of fiber a day.
And when scientists actually analyzed their stool samples to look at their microbiomes, what did they find?
They found a thriving, incredibly diverse ecosystem, one that is perfectly adapted to extract maximum energy from fibrous plants. Specifically, these children had massive populations of bacteria from the genus Bacteroidetes, and exceptionally high levels of a specific microbe called Faecalibacterium prausnitzii.
Okay, I want to highlight that specific bacteria. Faecalibacterium prausnitzii. Why is that one so important?
Because it is one of the most prolific, efficient producers of butyrate in the entire human gut.
Okay, the butyrate we just talked about.
Exactly. Remember, butyrate is the primary fuel for the colon cells and a potent systemic anti-inflammatory. Because the children in Burkina Faso were eating so much complex fiber, they had double the amount of these short-chain fatty acids circulating in their systems compared to the European children.
Which brings us to the kids in Florence, Italy, the representatives of the modern Western diet. What is their biological reality?
The diet of the Italian children was heavy in animal proteins, refined animal fats, and simple, highly processed carbohydrates. Think refined pasta, white breads, and sugars.
The standard stuff.
Right. And crucially, their fiber intake was staggeringly low, mirroring the average Westerner who consumes maybe 10 to 15 grams of fiber a day.
Which is what, half of what they should be getting?
Roughly half of the absolute minimum recommended amount, yes.
And what was the microbial consequence of that diet for the Italian kids?
Their microbiomes were depleted. They lacked the robust diversity seen in the Burkina Faso cohort, and terrifyingly, they were almost completely missing Faecalibacterium prausnitzii.
Wow, missing entirely.
They had starved off the very microbes responsible for maintaining the gut barrier and preventing inflammation.
This is the missing link. We look around at Western societies and see skyrocketing rates of obesity, type 2 diabetes, inflammatory bowel disease, and autoimmune conditions.
It's an epidemic.
And we try to blame it on a single villain, right? Too much fat, too much sugar, not enough cardio. But it all traces back to the soil of our internal ecosystem. We are chronically starving the organisms that protect us from these diseases.
Precisely. Chronic disease is often just the macro-level expression of a micro-level ecological collapse.
Okay, let's assume for a moment that you, the listener, take this to heart. You go to the grocery store, you buy the vegetables, you start feeding your microbes the structural fiber they need.
Great first step.
Once this bacterial army is fully fed, robust, and occupying the colon, how exactly do they protect us? Because we mentioned they prevent leaky gut, but their role as a defense system is far more aggressive than just acting like a patch on a wall, isn't it?
Oh, absolutely. Their role in pathogen defense and detoxification is highly dynamic. To conceptualize this, you have to realize that mathematically, your gastrointestinal tract is technically outside of your body.
Wait, what? It's literally in the center of my torso.
Geographically, yes. But biologically, imagine a human being as a very complex donut.
A donut, okay.
The hole in the center of the donut is your GI tract. It is a single continuous hollow tube running from your mouth to your rectum. Whatever is inside that tube has not actually entered your internal biology until it crosses the intestinal wall and enters the bloodstream.
That is a wild way to look at it, but it makes perfect sense. So you send a meal down the donut hole, a burger, a salad. Maybe you accidentally ingest some weird bacteria from an unwashed piece of lettuce. What happens as it travels down the tube?
The beneficial bacteria act as an intelligent microscopic filtration barrier. Their job is to extract the good stuff, the antioxidants, the vitamins, the amino acids, and facilitate their transport across the wall into your blood.
Okay.
But equally important, they ensure that the things you don't want stay trapped inside the tube to be excreted as stool.
So they're sorting the mail.
Sorting the mail perfectly.
And they are so good at trapping the bad stuff that researchers actually refer to the gut microbiome as a second liver.
Is a very apt title. The liver is your primary detoxification organ, responsible for filtering blood. But your gut bacteria neutralize a massive amount of environmental toxins before those toxins ever get a chance to reach your liver. Let's look at a specific mechanism involving endotoxins.
Let's do it. Break it down.
There are certain pathogenic gram-negative bacteria that possess a structural component in their outer membrane called lipopolysaccharide, or LPS.
LPS.
Yes, LPS is a highly potent endotoxin. If LPS crosses your gut barrier and enters your bloodstream, your immune system recognizes it as a severe systemic infection and triggers massive, full-body inflammation.
Like a five-alarm fire.
Exactly. It causes fatigue, joint pain, brain fog, and insulin resistance.
So LPS in the blood is very, very bad. How do the good microbes stop it?
Beneficial bacteria can actually physically bind to LPS molecules inside the gut lumen. They trap the endotoxin in the tube, preventing it from interacting with the gut wall, and escort it out of your body via bowel movements.
They just grab it and carry it out.
Furthermore, emerging science suggests that robust populations of beneficial bacteria can bind to and neutralize heavy metals and even carcinogenic substances formed when charring meats.
So if I take a course of broad-spectrum antibiotics and wipe out my good bacteria, I haven't just killed my digestion. I have actively dismantled my primary defense against endotoxins and carcinogens.
Exactly. When you decrease your good gut bacteria, all of that LPS, all of those toxins, now easily cross the compromised gut wall.
Because there's no one there to catch them.
Right. They rush into your portal vein and head straight for your liver. You are directly increasing the detoxification workload on your actual liver, which can rapidly lead to liver stress, non-alcoholic fatty liver disease, and systemic toxicity.
Let's talk about how they handle active invasions, because binding to toxins is one thing, but we swallow live viruses and aggressive pathogens all the time.
Every day.
How do the good microbes fight off the bad microbes? Is it just a matter of taking up all the physical space?
Taking up space is a huge part of it. We call that competitive exclusion. Think of the intestinal wall like a massive parking lot.
Okay.
If every single parking space is occupied by a beneficial, fiber-fed bacteria, an invading pathogen arrives and has nowhere to park. If it can't attach to the wall, it can't replicate, and it eventually just gets flushed out of the system.
I like the parking lot analogy, but it's not just passive, right? They aren't just sitting in the parking space aggressively staring at the pathogen.
No, they engage in active, brutal warfare, both physical and chemical.
Let's hear about the physical warfare first.
Okay, on the physical front, let's look at flagella. Many bacteria possess flagella, which are microscopic, whip-like appendages used primarily for movement,
Like little tails.
like little tails. But recent imaging has shown that certain beneficial strains use their flagella offensively. When a deadly pathogen, like a rotavirus, which can cause severe gastrointestinal rot and diarrhea, enters the space, the good bacteria literally whip their flagella around.
Wait, really?
Yeah, tangling up the virus and physically stopping it from moving or attaching to host cells.
They're essentially throwing microscopic nets over the invading viruses. That is phenomenal.
Now, what about the chemical warfare?
That is even more sophisticated. Beneficial bacteria communicate with the cells of your intestinal lining, stimulating them to produce antimicrobial peptides, or AMPs.
Explain AMPs. What is the mechanism there? Think of AMPs as specialized molecular torpedoes.
Torpedoes, I love it.
When a pathogenic bacteria tries to colonize the gut, the host cells release these AMPs. These peptides are positively charged, and the membranes of pathogenic bacteria are typically negatively charged.
So they attract each other.
The AMPs are drawn to the pathogen like a magnet, and when they make contact, they physically punch holes in the pathogen's cell membrane. The pathogen's internal contents leak out. It dies.
And the good bacteria are the ones coordinating this artillery strike.
Yes, they act as the reconnaissance team, signaling the host cells when to fire. And this coordination extends deep into the human immune system, specifically through structures called Peyer's patches.
Peyer's patches. I've heard this term thrown around in immunology circles. It sounds like something a pirate would wear. What exactly are they, and why do our microbes care about them?
Peyer's patches are specialized, localized clusters of lymphoid tissue located in the mucosal membrane of your small intestine. Think of them as immune system training camps embedded directly in the gut wall.
Training camps.
Yeah, they are packed with white blood cells, specifically T cells and B cells.
The heavy hitters of the immune system.
Exactly. The gut bacteria are constantly interacting with the surface of these Peyer's patches. It is a dynamic sparring session. The bacteria expose the immune cells to various microbial fragments, which trains the T cells and B cells on exactly what biological markers to look for.
Oh, I see.
When a real threat arrives, these immune cells are released from the Peyer's patches, fully trained, to actively patrol and protect the intestinal barrier from infection. Without the constant communication from the gut microbiome, these immune training camps would sit empty and uncalibrated.
Okay, so the microbes are the guards on the wall, they are the chemical artillery commanders, and they are the immune system drill sergeants. That alone justifies calling them a second organ.
Without a doubt.
But we haven't even touched on their most complex wizardry. And this is the part that fundamentally alters how we have to think about eating. The hidden enzymatic engine.
This is where we see the absolute peak of host microbiome synergy. The realization that human digestion is largely an illusion.
An illusion? Explain that. Because, I took high school biology. I learned that when I eat a piece of bread, the salivary glands in my mouth release an enzyme called amylase to break down starches.
Correct.
My stomach releases proteases to break down proteins, and my pancreas releases lipases for fats. We have our own human digestive enzymes.
We do, but our human enzymatic toolkit is pathetically small compared to the complexity of the natural world. Yes, human amylase can break down simple starches like a potato or white rice. But what happens when you eat an artichoke?
Or almonds, yeah.
Or a handful of raw almonds, or a bowl of lentils? The structural carbohydrates in these foods are built with incredibly complex chemical bonds. Your human DNA literally forgot to pack the molecular scissors required to cut those specific bonds.
So without external help, those incredibly nutrient-dense foods would just pass right through me. I wouldn't extract the vitamins, the energy, or the minerals.
Exactly. They would be completely biologically useless to you. But this is where the microbiome steps in. The bacteria residing in your colon possess a vast array of carbohydrate-active enzymes.
What are they called?
Specifically, families of enzymes known as glycosyl hydrolases, or GH, and polysaccharide lyases, or PL.
Glycosyl hydrolases. Those are the missing molecular scissors.
Yes. The human genome contains only a few dozen genes for digestive enzymes. The collective genome of your microbiome contains tens of thousands.
Wow.
When that complex artichoke fiber reaches your colon, the bacteria deploy these highly specific GH and PL enzymes to cleanly snip the complex chemical bonds apart, liberating the trapped nutrients, synthesizing vitamins like vitamin K and various B vitamins, and making them bioavailable for your human cells to absorb.
It is unbelievable. We are entirely dependent on foreign microbes to unlock the nutritional value of our own planet's flora.
100%.
But the sources we reviewed didn't just stop at breaking down plant fiber. They detailed how these microbial enzymes actively dictate our fat metabolism. This totally blew my mind. Walk me through the mechanism of bile salt hydrolase.
This is a perfect example of how microbes control human physiology at a cellular level. When you eat fat, your liver produces primary bile acids. These acids travel into the intestine to act like soap, emulsifying the dietary fats so you can absorb them.
Okay, standard human digestion so far.
But once those primary bile acids have done their job, the gut bacteria step in. They possess an enzyme called bile salt hydrolase, or BSH. The bacteria use this enzyme to modify or deconjugate the primary bile acids, turning them into what we call secondary bile acids.
Like what?
Such as lithocholic acid.
Why do we care if the bile acid is primary or secondary? What does a secondary bile acid do?
It acts as a master key for your cellular metabolism. These secondary bile acids absorb into your bloodstream and travel to various tissues in your body. When they arrive, they seek out and bind to specific receptors on your human cells, primarily receptors known as FXR and TGR5.
Let's pause and translate the alphabet soup for a second. What is FXR? What is a receptor doing here?
Think of the FXR receptor as a thermostat located on the wall of your human cells. When the microbially modified secondary bile acid floats by and bumps into that thermostat, it clicks it over.
Okay.
That click sends a signal to the nucleus of your cell, commanding it to regulate fat storage, increase energy expenditure, and decrease systemic inflammation.
Wait, so you are telling me that the biological signal that tells my body to burn fat and balance insulin doesn't originate from my human brain?
No, it doesn't.
It originates from a bacterial enzyme modifying a liver acid in my colon.
Exactly. Without the bacterial BSH enzyme making that specific modification, the thermostat never gets pressed. The signal is never sent.
That is massive.
It is. This is why researchers are finding that dysbiosis, a broken microbiome, is a massive contributing factor to metabolic syndrome and obesity. Your body fundamentally loses the ability to properly metabolize and store fat because the bacterial signaling molecules are just missing.
That completely reframes the obesity epidemic. But it gets even crazier. They have the molecular scissors for complex carbs, they have the thermostat keys for fat metabolism, and they also dictate protein breakdown.
Yes, they do.
And this is where the gut connects directly to the brain. Let's talk about neurotransmitters.
The gut-brain axis is perhaps the most heavily researched frontier in medicine right now. Microbial enzymes are responsible for breaking down undigested proteins into essential amino acids, things like tryptophan, valine, and leucine.
Okay.
But it's what they do with these amino acids that is truly profound. Let's look at a specific amino acid called glutamate.
Glutamate, you find that in lots of high-protein foods, right?
Yes, it is very common. When glutamate reaches the gut, specific strains of beneficial bacteria use their unique enzymes to convert that glutamate into gamma-aminobutyric acid, universally known as GABA.
GABA. For anyone who has ever looked into sleep supplements or anxiety medication, that is the holy grail.
It is. GABA is the primary inhibitory neurotransmitter in the human central nervous system. Its entire job is to calm the nervous system down, reduce neuronal excitability, alleviate anxiety, and promote deep restorative sleep.
But hold on, if the bacteria are producing GABA down in the colon, how does it affect my brain? It has to cross the blood-brain barrier, doesn't it?
It doesn't necessarily have to cross the barrier to have an effect. And this is where the physical wiring of the body comes into play. You have a massive neural superhighway connecting your gut directly to your brain, called the vagus nerve.
The vagus nerve.
Yes. When the gut bacteria produce GABA, those molecules interact with the enteric nervous system, the vast web of neurons lining your gut. This stimulation sends electrical and chemical signals straight up the vagus nerve to the brain, effectively telling the brain to calm down and reduce anxiety.
This is a total paradigm shift for mental health. I mean, imagine someone who is suffering from severe, unexplained anxiety or chronic insomnia.
This is so common.
They go to a psychiatrist, they get put on SSRIs or anti-anxiety meds because everyone assumes it's a chemical imbalance in the brain. But what if their brain is perfectly fine?
What if the gut is broken?
Exactly. What if the actual issue is that they took 3 rounds of antibiotics last year, wiped out the specific bacteria responsible for enzymatically converting glutamate into GABA, and now their gut is simply failing to send the calm down text message to their brain?
That is exactly what clinical researchers are exploring. A compromised microbiome leads directly to a compromised neurotransmitter profile. We are learning that many psychiatric and cognitive issues are, at their root, enzymatic issues occurring in the large intestine.
If they control our natural chemistry to this degree, what does this mean for artificial chemistry? Like pharmaceuticals. I take a pill, I assume my liver processes it. But if my gut is a massive chemical processing plant, how does my unique microbial profile affect my medication?
It dictates everything. This is the foundation of personalized medicine. The microbial enzymes in your gut have the power to structurally transform medications.
Transform them.
Yes. They can either activate them so they work, or inactivate them so they're useless, or worse, convert them into highly toxic metabolites.
Give me a real-world example of this.
The cardiovascular drug digoxin, which is used to treat heart failure. In a significant percentage of patients, digoxin simply doesn't work. For decades, doctors didn't know why.
And what did they find?
It turns out a specific gut bacterium called Egerthella lenta produces an enzyme that rapidly breaks down digoxin and inactivates it before it can ever reach the heart. If you happen to have a high population of this bacteria in your gut, the drug is useless to you.
So the doctor keeps upping the dose, try and get a response, not realizing the bacteria are just eating it. What about drugs where it becomes dangerous?
Warfarin is a classic example. It's a powerful blood thinner. The therapeutic window for warfarin is incredibly narrow, a little too much, and the patient can suffer severe internal bleeding. The metabolism of warfarin is heavily influenced by the gut microbiome's production of vitamin K and its specific enzymatic interactions with the drug. Because every single human has a wildly different microbiome, the efficacy and toxicity of this drug vary massively from person to person.
So in the very near future, a doctor practicing blindly will be considered negligent. They won't just write a prescription for a standard 50-milligram pill. They will have to sequence your stool, look at the exact enzymatic capabilities of your unique microbial workforce, and say, okay, based on your specific bacteria, this drug will turn toxic in your liver, so we have to use this other one.
Exactly. Pharmacomicrobiomics will become the standard of care.
Okay, we have outlined the sheer, undeniable power of the second organ. We know it maps out over our entire body. We know it requires complex fiber to prevent it from eating our own organs. We know it acts as an intelligent detoxifier fighting physical warfare. And we know its enzymes dictate our fat storage, our brain chemistry, and our medications.
We've covered a lot of ground.
We really have. But knowing all of this, we have to look at the grim reality. The modern world is a microscopic death trap. We are systematically destroying our ecosystems every single day.
We are experiencing what ecologists call mass dysbiosis.
Mass dysbiosis.
Yeah, dysbiosis is simply a state of severe imbalance, a catastrophic loss of beneficial microbial diversity, and a dangerous overgrowth of pathogenic opportunists. It is driven by overly sterile built environments, the systematic removal of fiber from ultra-processed foods, and the chronic, indiscriminate overuse of broad-spectrum antibiotics.
Both in medicine and in the agricultural animals we eat, right?
Exactly.
We are dropping biological nukes on our internal rainforests on a daily basis. So how do we stop the bleeding? How do we rebuild and fortify the fortress? Let's start with the internal environment. Diet. What is the tactical approach here?
The foundational step is reintroducing dietary fiber. But there is a massive caveat that catches most people off guard. You must do it gradually.
Why gradually? If fiber is the superfood, why shouldn't I just go eat 3 bowls of lentils right now?
Because of what we discussed earlier, extinction. If your microbiome has been depleted by years of a Western diet, you currently possess a very small population of fiber-fermenting bacteria.
Okay, I see where this is going.
If you suddenly drop 40 grams of highly complex fiber into your colon, that tiny workforce cannot process it efficiently. They will become overwhelmed, they will ferment the fiber abnormally, and they will produce massive amounts of hydrogen and methane gas. You will experience severe bloating, cramping, and gastrointestinal disgust.
And that's exactly when people quit and say, see, vegetables are bad for me.
Exactly. You have to slowly rebuild the workforce. Start by incrementally increasing prebiotics. Prebiotics are specific types of fibers that selectively feed the most beneficial strains.
What should I be putting in my grocery cart?
Look for foods rich in a prebiotic fiber called inulin. Asparagus, leeks, raw garlic, and onions are incredible sources. By introducing these slowly over weeks and months, you trigger the replication of the good bacteria. You're essentially building the army back up cell by cell.
Okay, so prebiotics are the food for the microbes. What about probiotics? Actually ingesting the live bacteria themselves. Should I just buy a $50 supplement pill?
Supplements can be useful in clinical scenarios, but evolutionarily, humans got their probiotics through fermented foods. Things like high-quality unsweetened yogurt, kefir, unpasteurized sauerkraut, and kimchi.
Why are fermented foods superior to just eating raw vegetables?
Because of the enzymatic predigestion we talked about earlier, when you ferment cabbage to make sauerkraut, the live bacteria in the jar have already begun the enzymatic breakdown process. They have synthesized vitamins, produced organic acids, and broken down complex cellular walls.
So they did the hard work for you.
Yes. When you eat it, the nutrients are immediately highly bioavailable. It takes a massive strain off your own compromised digestive system, while simultaneously delivering a massive payload of live, transient microbes that help reacidify the gut and suppress pathogens.
But let me challenge this. If I've had a bad diet for 10 years, and my gut is currently dominated by bad, pathogenic bacteria, do they just surrender when I eat a bowl of sauerkraut? Do they just pack up and leave?
Absolutely not. Pathogenic bacteria are ruthless survivors. When they colonize a gut, they establish defensive structures called biofilms.
Biofilms. What is that mechanically?
Imagine a bacterial colony building a microscopic, slimy, impenetrable dome over themselves. This matrix of extracellular DNA and proteins acts as a physical shield. It protects the bad bacteria from your immune system's white blood cells. It protects them from antimicrobial peptides, and it even shields them from pharmaceutical antibiotics.
Wow.
Many chronic recurring infections are driven by bacteria hiding safely inside these biofilms.
So how on earth do we breach the dome?
Through sustained competitive pressure and chemical alteration of the environment, you consistently eat a wide variety of plant fibers and polyphenols, the colorful compounds in berries and vegetables, and introduce the organic acids from fermented foods, you slowly begin to alter the pH of the colon.
Making it inhospitable for the bad guys.
Exactly. This shift in acidity, combined with the beneficial bacteria crowding the space, begins to chemically degrade and dissolve those pathogenic biofilms. Once the dome is compromised, your immune system can rush in, clear the bad bacteria, and allow the good bacteria to repair the mucosal wall. It is a slow, methodical siege.
It's not a quick fix. You are orchestrating a long-term ecological restoration. Now, what about the outside of the fortress? We talked earlier about how overwashing destroys the skin's acid mantle and kills protective strains like Staphylococcus epidermidis. What is the actual protocol for skin hygiene?
We need to fundamentally regress our hygiene practices. The most actionable step is to completely banish harsh antibacterial soaps from your daily routine. Unless you work in a surgical ward or are dealing with a specific pathogen exposure, you simply do not need them.
But people want to feel clean. What should they use?
Use mild pH-balanced non-bacterial soaps and use them incredibly sparingly. Focus only on the apocrine gland areas, the areas that truly generate odor, like the underarms and the groin.
And the rest of the body.
For the vast majority of your body, your arms, your legs, your torso, warm water and mechanical friction with a washcloth is more than sufficient to remove dirt and dead skin without indiscriminately nuking your protective microbial rainforest.
Which brings us to the water itself.
Yeah.
Because it's not just the soap that's killing the microbes, is it?
No, it is the municipal water supply. Most modern tap water is heavily treated with chlorine or chloramine.
Which makes sense, right? We don't want cholera in the water supply.
Absolutely. Chlorination is one of the greatest public health triumphs of the 20th century. But we have to look at the mechanism of chlorine. Chlorine is a powerful oxidizing halogen. Its entire biological purpose is to instantly tear apart the cell walls of bacteria on contact.
It doesn't discriminate.
Exactly. It doesn't discriminate between cholera and the highly beneficial lactobacillus on your skin. When you stand in a hot, steamy shower for 15 minutes every day, bathing in and inhaling vaporized chlorine, you are subjecting your skin and respiratory microbiome to a daily chemical assault.
So what's the solution?
Installing a high-quality shower filter that specifically neutralizes chlorine and chloramines is one of the easiest, most passive interventions you can make to protect your external ecosystem.
What about personal care products? Lotions, deodorants, perfumes?
Look at the ingredients.
Yeah.
Many commercial products use high concentrations of alcohol as a quick-drying base or a preservative. Alcohol is a potent biocide. It desiccates and kills microbes. Transitioning to natural oil-based products or essential oils can drastically reduce the daily chemical burden on your skin flora. And there was a highly specific, very practical recommendation regarding bathroom hygiene in our sources.
Oh yes, the bidet.
It sounds trivial, but it is ecologically significant.
Yeah.
The perianal and genital regions host incredibly delicate specific microbial populations. Using dry, rough toilet paper, or worse, chemically treated flushable wipes laced with preservatives and fragrances, completely disrupts this localized flora.
Makes sense.
A bidet allows you to cleanse the area with plain water, preserving the mucosal integrity and the fragile microbial balance of that specific biome.
It's incredible how all of these interventions point in the exact same direction. The irony is almost painful.
How so?
We spent the entire 20th century, hundreds of billions of dollars trying to sanitize our world. We invented antibacterial sprays, hand sanitizers, processed sterile foods. We equated complete sterility with ultimate health.
We did.
And now, the highest echelons of modern science are looking at the biological data and saying, we made a terrible mistake. The secret to health is to literally go outside, put your hands in the dirt, and eat bacteria-laden fermented cabbage.
It is a profound paradigm shift from eradication to cultivation. The epidemiological data is crystal clear. Populations that interact with their natural environment thrive. People who exercise regularly and sweat actually induce physiological changes in their gut that increase microbial diversity.
Really, just from sweating.
Yes. People who garden, who grow their own food, who have dirt under their fingernails, are constantly inoculating themselves with robust environmental microbes. We evolved in a microbial soup. We require constant exposure to maintain the calibration of our immune systems. Isolation and sterility are the enemy of biological resilience.
Okay, let's pull all these threads together. This has been a massive journey. We started by realizing that human beings are essentially walking, breathing planets covered in wildly diverse, specialized ecosystems. We looked at the skin, where fungi like Melissetia live on our oils and bacteria maintain our protective acid mantle. We learned about the critical stakes of day one, where vadbital microbes calibrate an infant's entire immune system to prevent a lifetime of allergies. We traced the path down through the acid vat of the stomach into the colon, where a trillion-strong army desperately waits for structural fiber.
And we learned that if we fail to feed them that fiber, they will literally consume the mucosal lining of our own organs, leading to leaky gut and chronic systemic inflammation.
But if we do feed them, they act as an impenetrable defense force. They act as a second liver, binding to endotoxins like LPS and flushing them away. They physically tangle up deadly rotaviruses with microscopic whips, and they coordinate chemical artillery strikes by commanding our host cells to release antimicrobial peptides.
And beyond defense, they act as our primary metabolic engine. They deploy specific molecular scissors to extract energy from plants. They modify our bile acids to click the cellular thermostats that control our fat storage. And they enzymatically synthesize neurotransmitters like GABA to calm our central nervous system via the vagus nerve.
It is a masterpiece of evolutionary biology. We are entirely dependent on them. But as we wrap up this deep dive, there is one final piece of data that really forces you to step back and reevaluate what it actually means to be human.
Yes, the genetic comparison. When we sequence the human genome, we find that at the level of our host DNA, human beings are incredibly similar. You, me, and every single person listening to this right now, we are roughly 99.9% identical in our human genetics.
99.9%. That is a vanishingly small margin to account for all the massive variations we see in human health, metabolism, and immunity across the globe.
Exactly. But when we look at the metagenome, the collected genetic makeup of the individual microbiomes we carry inside us, the numbers flip completely. Your specific microbial genetic fingerprint can be 80% to 90% different from mine.
Let that sink in. 80 to 90% different.
So if our human genetics are practically identical, but it's our highly variable microbiome that actually drives our daily digestion, dictates our fat storage, calibrates our immune resilience, synthesizes our neurotransmitters, and even determines whether a medication will cure us or kill us. It forces a very uncomfortable, profound philosophical question.
Which is?
Are we autonomous human beings who happen to be hosting a microbiome? Or are we, in evolutionary reality, just highly advanced bipedal transport vessels built specifically to feed and carry trillions of microscopic life forms?
That completely shatters the ego. We aren't the main character. We are just the planet they live on, the vehicle they drive. Think back to that half-gallon container of invisible creatures we talked about at the very beginning of this deep dive, the one weighing down your biology. The next time you sit down to eat a meal, or reach for a harsh soap in the shower, or feel a wave of unexplained anxiety, remember the sheer scale of the ecosystem you are managing. You aren't just making a lifestyle choice for one organism. You are dictating the survival of 100 trillion of them. The X-ray machine might not be able to see them, but they are there. They hold the molecular scissors, they hold the cellular keys, and they hold the line between chronic disease and vital health. They are doing the work. Make sure you feed the crew.